Hi, welcome to the third of these short videos. This one might be a little bit longer, so I've left on the aircon. There might be a little aircon fan noise, but hopefully that won't be too bad. So in this, I want to introduce the idea of inferring evolutionary history, which is, of course, a major project in phylogenetics. It's one of the main things that we do. So what we want, of course, is to model phylogenetics, to model that process, the process from ancestor to the current reality, if it's a species or a gene family. And of course, we know that mutations in the genome will change the DNA. They create new alleles and they change the frequency of alleles in the population. And one characteristic is that changes that give an advantage in selection may become fixed. So, of course, there's a lot to look at in terms of basic evolutionary biology and so on. And we're not going to do that because we're really going to be working on just the bioinformatics of this from the biology perspective. But nonetheless, it's good to have an idea of the forces at work. So there's negative selection, which we'll discuss. There's drift or neutral evolution, and there's positive selection. So these are kind of the negative and negative to positive axis of forces. And then there are these specific events like genetic expansion. So that would be duplication, gene duplication, other duplication events. Deletion, so we can see a region get deleted instead of duplicated. And we can also see just new genetic material introduced not by inheritance, but by other means. And that's referred to as horizontal transfer. So let's look at those a little bit. So positive and negative selection are the two major topics that people first think of when they think of evolution. Actually, probably most people will think of Darwinian selection as positive selection. That is the process by which new advantageous variants sweep and become fixed in a population. So this is also called molecular adaptation. And it's usually what people think of when they think of Darwinian evolution at the DNA level. But negative selection is a thing too, and it's a common phenomenon. And this is another form of natural selection, but instead of selecting for something new, for introducing something, it's selecting against change. So if change is introduced and that change is deleterious, there will be selective pressure against change. And this is the outcome of this is that the process preserves functionally important sites. So it works just the same way evolution works at the molecular level. We have random mutations in DNA that are created by the usual process. However, if these occur at functionally important sites, they will have negative consequences for fitness of the gene or the organism. And then as a consequence, the sequences at those functionally important sites will be conserved and will change less often. We've already seen this in things like multiple sequence alignment, where we see motifs, functionally important motifs, zinc fingers, heme binding motifs, and so on. Those residues don't change because they are required for that function. And the preservation of the function is an impediment to molecular change. In the middle, between negative and positive selection, is drift. And drift is not itself new, but there was a proposal in the 1960s by Kimura, which was that most mutations would be selectively neutral, and that they could then be fixed in the population. And so the consequence of this model was the idea, in principle, that perhaps most of the fixed change that's observed would be from genetic drift rather than natural selection. This is referred to in general as the neutral theory, and it was a big shakeup in molecular evolution in the field in the 1960s. And there were arguments in the 1970s and so on about whether it was compatible with natural selection. It is, but the emphasis is elsewhere. So the emphasis is less on positive and negative selection and more on neutral drift. The broad predictions of the theory, I believe I'm not an expert in this, but my understanding is that the broad predictions of the theory have not held up to formal tests. So one can test the predictions of ancient gene frequencies or allele frequencies, and in those attempts to test neutral theory, it hasn't held up so well. as theory, but nonetheless, neutral uh, neutral change drift is extremely important. And in fact, one of the really positive outcomes of this is that it gives us a rationale for understanding when neutral change happens. We might not expect that change to happen any faster by positive selection or slower by negative selection than just the change, the underlying process itself, the underlying mutational process. And because of that, neutral change might be expected to lead to something that's been referred to as a molecular clock, where the rate of change, of neutral change, is kind of an estimate for the amount of evolution that is going on. So we can use that as a principle to observe selection. So we have to make a bunch of assumptions in this, but let's make those assumptions and see where it takes us. So let's assume first that some changes are not subject to selection. So they're not going to be subject to negative selection or positive selection. Let's take that further. Let's assume that the changes that are subject to selection in coding sequences are going to be changes in amino acids, so that changes that don't change the amino acids are not going to be subject to selection. Now, there are lots of reasons where that might not be strictly true, but it might be a reasonable model in many, many cases. So what that means is that changes in amino acids, non-synonymous mutations, are more likely to be subject to selection than synonymous changes. And say we also have some measurement, we devise some measurement when we're looking at a bunch of different changes to estimate the rate of change from a set of observations of change. And if we can estimate that rate, and we can estimate that rate separately for the synonymous changes that don't change the amino acids and the non-synonymous changes that do, we can compare those rates. And selection in those protein coding regions can then be inferred by the ratio. So if the synonymous mutation rate, the rate that doesn't change amino acids, is not subject to selection, which is this first bullet point, then that rate provides an estimate of the neutral change rate. And then the non-synonymous mutation rate can tell us whether the non-synonymous changes are either faster or slower than synonymous changes, reflecting either positive or negative selection. So let's hold that thought and go back to 1969 and address a question that was an open question in 1969. And that is about the origin or the lineages that led to modern humans. So it was known that humans and old world monkeys and apes had a common ancestor relatively recently in evolutionary history, but it was not entirely sure, it was not entirely settled whether what the order of branching was. So it was known that there was a common ancestor some period ago, but it wasn't known, for example, that humans and apes diverged more recently from a common ancestor than the common ancestor of humans and apes did from old world monkeys. So there were a number of researchers who worked on this problem, but I'm going to focus on this paper by Wilson and Sarek. What they did was a series of measurements of that sort of evolution, of those sorts of differences. They used three methods. They used microcomplement fixation for proteins, which is an immunological measurement of differences. They used DNA hybridization, the experimental DNA hybridization, and then they used a bioinformatics technique, which is basically sequence difference between them. So let's look first at the microcomplement fixation on the left two. This is albumin and transferrin, and they compare humans and the great apes, and they compare humans and old world monkeys, and they see that the difference between humans and old world monkeys is much larger than the difference between humans and the great apes. The same is true for transferrin. So both of these protein-based measurements show that larger difference. The same is true when they do DNA hybridization between the DNAs of these different species. And the same is true when they look at the sequence differences among the globins. Remember, this is prior to the invention of DNA sequencing, so they were looking at protein sequences. And the globins were among the sequences that were most highly sequenced. So all show consistently that the humans and the great apes are more similar to each other than humans and old world monkeys, which then favors this model on the right, over the model on the left, or an alternative model. That doesn't use any information about synonymous and non-synonymous mutations that we mentioned a couple of slides ago, but I asked you to hold that thought. So it is the case that the degeneracy of the genetic code means that there will be synonymous and non-synonymous mutations, right? There will be changes that do not lead to changes in proteins, and we can observe those in the same coding sequences where we observe amino acid changes. There are also other differences. So in DNA mutations, transitions and transversions, as we'll see shortly, also occur at unequal rates. Different processes that contribute to mutations leave different sorts of signatures that leave different rates of mutations, but we're going to ignore that for the moment. But we've already seen that positive selection can be inferred if we see an elevated rate of non-synonymous mutations over the rate estimated from a neutral model. So that was 1969. Let's jump forward to 2003. And so here we're looking at humans and chimpanzees. So that's one of the great apes, and we're looking at humans and chimpanzees. The DNA sequences are nearly 99% identical. We still wind up using this. So in this case, what they did is they looked at gene sequences of 7,645 chimpanzee to orthologs in human and in mouse. So these are all orthologous sequence comparisons. And they used this 3-species sequence alignment, and then they looked at the estimate of the rates of mutation in each of those. So they end up having 7,645 comparisons, 7,645 models, and as you'll see on the next slide, they use that to estimate where the human accelerated genes, that is, where does positive selection occur on the lineage that leads to human. And this is by fitting models that include parameters specifying rates of synonymous and non-synonymous mutations. So this is the basic model. Here we have one hypothesis on the left and another hypothesis on the right. So that's H0, our null hypothesis. And we have some rates. So here the rates are D sub S and D sub N. These are the estimated rates of synonymous and non-synonymous mutational change. There are several different allowances here in this hypothesis 0. So in the 0 hypothesis, we either have, in the null hypothesis, we either have negative selection, so the non-synonymous change rate is less than the synonymous change rate, and that's less than 1, or the non-synonymous rates are equal, and that's neutral selection. Here, this means mouse, this means human, and this means chimpanzee. So here we see the common ancestor of mouse, human, and chimpanzee. And so we're looking at the rates along each of these branches. Now, the alternative hypothesis is that, in particular, on this one branch, the rate of non-synonymous change is larger than the rate of synonymous change. So these are the two hypotheses that are compared, and we're allowing for this different possibility in this alternative model. Now, there's no reason to go through all of the details of this. Suffice it to say that they set this up so that this model could be tested, and they tested it on each of the 7,645 genes. And they found some of those genes where there seemed to be an acceleration, where this alternative hypothesis was preferred, meaning that there was an evident acceleration or positive selection along this branch that leads to humans. When they identify those genes, they can identify then characteristics of the functions of those genes. And so what do they see? So here they're looking at a method we haven't gotten into yet, but we'll get into to look at the gene ontology, to look at the annotation of this. And basically what they identify is a set of biological processes that appear accelerated in the human, the lineage that leads to human. And so here we have the human lineage olfaction. Okay, so this means this is a p-value of this is a very low p-value, and we see 48 genes that appear to be accelerated in the human lineage that involve olfaction. So olfaction, sensory perception, cell surface mediated signal transduction, chemosensory perception, a lot of events here that involve sensation, certainly, olfaction, sensory perception, hearing, and so on. You know, these sensations really do make a difference. So just the Nobel Prize was awarded just last week to scientists for the discovery of hot and cold and pressure sensation, molecular, you know, kind of a molecular description of that. So this is kind of what we see. We see these processes that appear to be accelerated in the movement towards the human lineage, and that tells us something about the kinds of processes that make us, you know, distinct from our nearest living relatives, the chimpanzees, despite having 99% sequence identity at the DNA level. So that is where I'm going to leave it for this video, and this is the third of the series of videos that are introducing phylogenetics to students in LSM2241 Introductory Bioinformatics.